Well, good morning, afternoon, evening, everybody. My name is Jack Deckers. I'm a professor at Iowa State University in Department of Animal Science, and I was one of the co-PIs of the AG2PI grants that we received from NIFA, which we're very appreciative of. And no, they've ended. That funding has ended. But we still have, as part of that grant, those grants, we distributed a lot of the funding through seed grants and then later on larger seed grants, which we nicknamed coconut grants. And so we're using the opportunity here to give recipients of those larger grants, the coconut grants specifically, to talk about their progress and what they've achieved and getting some discussion going. So that's the purpose of this field day and also the next two field days that we'll have. So today we have presenters from Texas A&M and Oregon State University. And we're going to start with Seth Murray, Mahendra Bandari, and Jose Landovar from Texas A&M, and they're going to talk about the use of drones in plant breeding and data collection. So as usual, please put questions in the chat, and I'll keep an eye on the chat. And if there's a question that is immediately relevant, I'll interrupt the speaker. But otherwise, we'll leave them to the end. So Seth, floor is yours. You're muted.
Yeah, you got unmuted. Okay. Well, thank you, Jack, and thanks a lot to AG2PI and USDA NIFA for helping support this project. We're talking about facilitating community, and community is really important in using UAS or drones. Now, Mahendra and I were the co-PIs on this project, but there were lots of people, as you'll see shortly. And then there were a number of staff that I show pictures of here that you'll either hear from or will be discussed in this talk. And I really tried to balance, the reporting aspect of AG2PI getting a coconut grant, because it was really useful, but also talk about some science and new applications that this has enabled. One of the keys to this project, though, was trying to support the community. And this is just a list of a number of different groups, and it's certainly not meant to be all-inclusive, that do have activities in drone phenotyping, UAS phenotyping, from multi-state projects to the AG2PI group that you're hearing from now, to Phenome Force, which provides great webinars, a journal, North American Plant Phenotyping Network, and then traditional scientific societies. So I'll cover a couple of these as we go through, but I would encourage anybody who wants to learn more about UAS activities to get involved in these groups. And part of what we were trying to do is coordinate activities and communicate activities that were going on between many of these groups. So this is a list of some of our collaborators on the project when we initially proposed it. And if there's people that we're missing, we'd love to know about them. But I think all those groups I just showed, there's somebody representing most of those. Livestock, Joshua Jackson is representing that. It seems like the use of UAS in livestock is a little bit smaller than the use of UAS in plants right now. But that's a discussion we hope to facilitate going forward. I put in coordinator Anna Porter. She was hired sort of a little more than halfway through this project, and it's part of her role to help communicate these types of activities and facilitate them. So one of the early on sort of successes and things we did for this project that really surprised me in terms of demand was supporting some early career researchers, mostly graduate students and postdocs, to come to the S1069 multi-state project. And the S1069 multi-state project involves people all over the country, people that are routinely involved, and they meet once a year to discuss UAS in agriculture. Now, this isn't specific to phenotyping. A lot of these folks are actually on the extension side or on the agronomy side, but this is a really interesting group pushing the boundaries of how UAS will be used and how it could be used for advancing agriculture at large. One of the other things we did recently, and we plan to do many more of these going forward, is host a sort of a discussion. We had 4 presenters, and Mahendra and Jose, who you'll hear from, was one of those presenters, and Ace Pugh and Jennifer Lakowick from Montana State, and then the University of Florida, Dr. Wang. And I think this was really useful because it talked about this new restriction on UAS that's being rolled out through federal government mandates to some extent, especially against DJI drones and even some of the software. Some institutions are having difficulty using that. And so having those discussions where we can figure out how other states and other groups are working around these was really, really productive. And so that's up on YouTube if anybody missed it. We're hoping to have a couple others. Data organization and storage, especially making that data available, is a perennially topic of interest. Open source software and hyperspectral UAS are the two others that people have identified in this community as ones we need to have these type of discussions on. But we really welcome any further discussions, anybody who wants to lead these activities or speak on these activities to facilitate the discussion. So I'm going to move a little bit more into the actual activities of using drones towards phenotyping and phenomics. But I think the importance of language is really important here. I'm seeing a lot of confusion when I discuss with people when we talk about something like phenomics versus phenotype, right? And the phenome I think of as the genome, and saying a single phenotype is part of the phenome is the same thing as saying a single functional marker is the genome. So the phenome is much more broad, and I think so far we've barely tapped into it. So it's the totality of all those plants' measurable characteristics. The other language issue we see a lot is with drones and UAS. So unmanned aerial system, unoccupied aerial systems, uncrewed aerial systems, all of these are used in the literature along with drones. And we found two examples where somebody published a paper fairly recently where they said, well, this has never been done before. When in fact it actually been done 5 years earlier. It's just when they did their literature review, they didn't search these other terms. So these rapidly developing areas, we expect to have a lot of confusion when it comes to language. Multidimensional scaling and principal coordinate analysis or component analysis is another example where things developed in different disciplines and there wasn't that communication and coordination, and so people reinvented the wheel. And that's why we think this community building exercise is really important, to be talking about these things and reaching out to people, not just in the US, but also throughout the world. So when it comes to phenotyping, one of the concerns that I hear a lot is that it's technology for technology's sake. So I want to run down some of the uses and some of the different perspectives people have on this. So the first, and the one that generated the most interest, say 6-7 years ago, was automating routine measurements. So instead of having students go out there and measure plant height, can we measure plant height from a drone or from a ground vehicle? And what we've found over time is they give different measurements, but the UAS measurement or the drone measurement tends to be more heritable and more repeatable. And so if our standard is comparing to human-based measurements, sometimes people leave a little disappointed, right? But that's only just scraping the tip of the iceberg. I think the more exciting thing is finding new signatures of eliteness. So the temporal aspects, being able to routinely collect this data like plant height and figure out patterns. I'm going to talk a little bit about temporal growth patterns and senescence in this presentation. I think phenomic selection, which is different from phenotypic selection, is probably one of the most exciting areas to me personally. I'm not going to be talking about that today, but that's generating a lot of interest both in industry and in academic research. Now, all these are a little bit contrasted with the use of AI and deep learning, where there may be less explainability or less understanding, but the ability to process far more data and get much deeper insights. So there's other groups that are discussing AI and deep learning, but I think that's only just beginning as well. Now, I think it's important to recognize, too, that this doesn't just apply to geneticists and breeders and researchers in the field. Anything we discover here is also useful for identifying stress signatures that farmers could use in management for figuring out when a crop is stressed or applying irrigation water. And then lastly, we've already identified a number of new phenotypes and mechanisms of biological importance to this work. So I think, this new frontier is really going to change biology as well as applied plant breeding and applied agriculture. So I'm going to just show a couple examples and then I'll go into the process. This was one of the recent ones, real nice application for us. We had identified some loci through a GWAS study probably 8 years ago and developed isogenic lines to validate those. Then when we grew them in the field, we happened to fly drones over them that year. And the thing that was so surprising is if we had only measured terminal plant height at the end of the season, we would not have been able to validate these loci as real in these genetic backgrounds. But what we found actually was that these loci are acting far earlier in growth than we had ever known before. And we were able to show significance under there in figure 4 at early growth stages, but not at those late growth stages where people traditionally take manual measures. And so this is interesting because they were actually discovered, again, in a GWAS population by using manual measures, but being able to be confirmed only by the drones, or at least, I guess we could have taken manual measures at those earlier time points, but it's pretty inefficient to be able to do so. You know, the other type of new insights this is providing is when you do connect this to genomic data and you're trying to map loci in a linkage or LD population, a lot of these loci are not detectable at later stages that are detectable at earlier stages, right? So that may get at some of our hidden heritability. And probably the reason for this is just additional noise and additional loci mask those early loci in development, such as that pink QTL in the first location. I think it's also important to note that using these temporal flights throughout time, we can interpolate days that we did not measure. Just as in genomics, if you have a certain marker set, you can interpolate the marker the marker state in between those two flight dates. So there's a lot of really interesting new methods that are being developed. And if you went to the North American Plant Phenotyping Network meeting last week, you were probably blown away like I was with all the diverse activities and insights people are finding. So one of the things this AG2PI project supported was for us to sort of standardize and apply a standard UAV processing pipeline. And the three folks to the right there are absolutely critical for this. I don't know how to do any of this stuff myself. I mean, they're so advanced in their processing. I do want to point out, though, that it's really important to consider the pipeline and realize there are many pipelines out there that can be used. So this is the pipeline we've decided to use. And now that we've stuck to it, if we were going to go back and use a different pipeline, I think there's concerns about how comparable that data would be. And so I'll show you when I talk about the Genomes to Fields project and the 20-some environments that have been processed. If we decide to change that pipeline, then we need to go back and reprocess everything, most likely, to get the same type of insights. We still have a lot of problems with drones. We see distortions and mosaics. We see issues with flights just for whatever reason, we have to throw them away because we can't make an orthomosaic, or they have hot spots, or we find extra rows in the field or less rows in the field. And so Mustafa, especially, his detailed placing of shape files and looking for these things is really important in quality control. So when we talk about shape files, we generate orthomosaics because the images under any one drone picture, it's hard to tell what's in there. So just like you make a panorama with your phone, you orthomosaic and stitch images together to create an image of the whole field. And then we overlay a shape file that we can extract meaningful information out of that. We found this was an early bottleneck. And so Stephen Anderson and I wrote some R code a while back that's turned out to be really useful for connecting to breeders' field books. So in my breeding program, we typically have about 17,000 plots. And so if we're going to make a shapefile for every one of those plots manually, that would be completely impractical. So automating this step was a big way to go. And there's newer software out there, some that's using some deep learning approaches to help identify those shapefiles. But this is a really critical step in the whole process. So we get the UAV images, we create the orthomosaic shapefiles, and then we do data processing. And I showed below before that the data processing we're primarily using is Felipe Matias and company's Field Image R. And this is just an example right here of some shapefiles that, although our software automatically places them, there is some manual shifting that's needed due to those distortions in the mosaic or due to some other problems in the field. And having somebody manually do that still is a bottleneck, but we're really thankful that Mustafa was able to be supported on this project to do such a great job. And so this is yet another example of what those shapefiles look like. So from Field Image R, we tend to get somewhere in the neighborhood of 36 vegetation indices if we're using RGB, or closer to 90 if we're using multi-spec. And what you get is the tabular data that I, as a plant breeder, and many geneticists are much more comfortable to handle. So going from that image set to data you can use, you can use deep learning or AI, or you can use a pipeline like I previously showed. So by taking temporal flights, we're able to look at these vegetation indices and monitor these over time. Now we tend to exclude the soil. So these are not confounded with canopy cover or any type of leaf area index. This is purely a measure of plant height. And we find that the raw image bands, the blue, red, and green, tend to not be as heritable. They tend to not be as predictive. But by calculating ratios, we eliminate a lot of those issues from lighting or from clouds and other issues that the drone may have had. And so we really like to use these vegetation indices as we go forward and compare plots and map loci and stuff like that. So I'm going to give you a couple case studies just so you have an idea of where we're finding use in this before I show how we've expanded this to other locations. So Aaron DeSalvio came in as a PhD student a few years back, and he, through some discussion, we had a project to measure southern rust and senescence. And so senescence is when the plant dies at the end of the season, which in Texas is usually sometime in July and August, as it's well over 100 degrees most of the time. So you can see here, we're making those routine drone flights throughout the growing season, and then we take a certain window to just look at senescence. So one of the things Alper identified, Dr. Adek identified, was this period of grain filling. And this period of grain filling is highly correlated to grain yield, as you can see in the bottom right. And so what that is simply a measure of the day that the plant flowered, which is its first ability to start moving photosynthates into the grain, to the day that it senesces, which is not the same as black layer maturity, but is much more easier to scale to thousands of plots. So that grain filling period there behaves differently for different genotypes. And it's actually a heritable and under genetic control trait that nobody's ever been able to measure before, or at least not measure practically without a lot of undergraduate field labor. A common concern is that a lot of these vegetation indices are confounded. What we've actually found, part F here at the bottom right, is genotyping by sequencing data for a certain population that we used in the study. And I hope what you can see is that these vegetation indices across time are no more multi-collinear than genomic data. So if you don't have concerns about collinearity in genomic data, you similarly shouldn't have concerns about calculating many vegetation indices over many time points. And of course, with machine learning approaches, we sort of minimize the overfitting that may occur from that. So the second objective of our AG2PI project was actually to take the drone data that's being collected in the Genomes to Fields project and start running it through a standard pipeline. And I showed you that pipeline and who's responsible for that. Now this has, Genomes to Fields project is such a huge project, there's no way to acknowledge all the many collaborators. But in our 10th year now in 2024, there's over 180,000 field plots, 2,500 corn varieties, and 162 unique environments, of which a proportion, probably 10 to 20% of them, have drone data. The other thing that's interesting about this Genomes to Fields project is that we're all collecting the same manual phenotypes. So we can connect any drone measurements directly to traits of interest, especially grain yield, plant height, days to flowering, things like that. Last year, there was a Genome to Field environment prediction competition where anybody could enter using the massive data set that's already been collected from this public sector project and predict the outcome of the current year, which in this case, I think was 2022. I think there were something like 200 entrants, and it was really impressive to see the variability between people's predictions. But this is a real data set now that is being used to help the whole community improve on prediction of N phenotypes, given genomic data in this case. Now this year, we're talking about, we don't know if it's practical to start incorporating some of the phenomic data. So under this project, this has basically been close to a year of Mustafa and Alper and Aaron and other people processing these Genomes to Fields data. We focused on 2020 and 2021 because the same hybrids were grown in those environments. And these were every location that we have data from that we could actually process and use. I think I want to draw your attention to a couple things. First of all, some groups collected multispectral data, some groups collected RGB data. The number of flights is radically different across here, from 8 flights in Nebraska in 2021 to 87 flights in Missouri, which included both RGB and multispectral. And when we start going over to the right-hand side and looking at the amount of data that some of these locations have, which depends on the sensor, how often they flew, how many, what height they flew at, we start to talk about substantial amounts of data to deal with. And that's why discussing data storage and making this data available is really an important priority going forward. So let me show you a little bit about how this multi-location data was used, but first focus on Missouri. So Jacob Washburn in Missouri and Sherry Flint Garcia and others collected G2F data, I think in 2020, that had those 87 flights. This is just the 40 flights of multispectral data, I'm sorry, RGB data. And one of the biggest insights from this is we now think that flying every 2 days actually is not even too much, right? We're getting new information every 2 days from these flights. And this is just representing plant height, but I think that's really applicable to the vegetation indices and potential AI and deep learning approaches. So I'm not expecting a company would apply this in a breeding process, but this is the type of insights we're looking for: is when is the right time to fly? How often is the right time to fly? And how can we make a better understanding of the data we have for biology? Just mainly wanted to highlight one thing in this. This is some of the locations of genomes to fields that drones were flown at. If you look in the center here, the flight dates don't match. So one of the things we've been working on a lot is how do we connect these different flight dates? For plant height, because you can model it easily, it's not as hard. But when we start talking about vegetation indices and new traits and phenomic prediction, this is a major challenge. And so functional principal component analysis and area under the curve analysis are some aspects we're currently wrapping up right now. A surprising insight that came, at least I think, is the rates of growth and how different they were between these G2F experiments. So this is a number of locations that were all flown with drones, and we have a temporal time course up until flowering. And you can see that, for instance, Texas, our late planting Texas trial rapidly increased height much faster than the cooler and more temperate environments, which probably is expected. But what's unexpected is how there was genotype and environment interaction, and also the dramatic difference that we saw across all these. So now with weather data and end traits like yield, we can connect all this and end up making better predictions, hopefully, of genetics and the end goal of breeding or whatever trait you're interested in. So my last focus slide here is something that's hot off the press. Aaron just sent this to me this morning. We now have 22,000 plots from across these G2F locations that have been scored for senescence by Cody Kettler and Luke Gray, and Nick Shepard, who's a new PhD student, helped Aaron with some of this deep learning approaches. And we were really happily surprised to see in this upper right confusion matrix how well the deep learning approach is predicting senescence. And the reason this is important is because it's really miserable and takes a lot of time to manually score traits from drone images. So now that we have a pipeline for this, the goal would be to deploy this among the probably hundreds of thousands of plots we've collected over the years for UAS data. So with that, I'm going to turn it over to my colleague Mahindra, co-PI on the project.
Thank you, Seth. I hope everyone can hear me okay. So Seth mentioned about helping the community. So we are part of another big project that we are helping to process and standardize the UAS data sets. And this is the WITCAP project that George from UC Davis is leading, and Dr. Amir Ibrahim from Texas A&M University is leading the UAS component of the project. And we work together with Dr. Gina Zhang, I think he's here as well, from Purdue University, to help, help all the wheat breeding programs on how to collect the data sets, what are the standard protocols to collect the data sets. And then we developed this WITCAP UAS hub where the breeding programs, once they are done collecting the data, using this hub can upload their raw images, and we receive those data sets, we download them, and we follow our pipeline and develop the orthomosaics and generate those vegetation indices and canopy features. And then through this same website, we send the data sets back to the breeding programs. Seth, if you can go back. So as you can see in this slide here, this project started in 2021, and we started getting the data from 2022. And in these couple of years, we have processed the data from almost 15, 16 breeding programs across the country. And we were able to deliver the data products, and the end goal to deposit all these phenotypic information is to transfer the data in the T3 database where the genomic database data sets are stored. So the project is looking forward on integrating those genomic data sets and all of these phenomic data sets working together with our team and the breeding programs. Next slide. Okay, so now again, coming back to helping the community. So what we thought is working in this project and with other breeding programs across the country, we realized that there are so many basic questions that the new beginners wants to learn from. For example, we get questions about what kind of sensors do I need to buy if I am a new beginner that wants to implement UAV program in the research program. What kind of sensors, what kind of software or things like that that we need to follow to get the data sets that we want to. And we thought, like, what if we develop a website that has all these informations that are well tested and put it together in a very easily understandable form. And that is what we proposed for this project. And we tried to develop a website on putting together all these informations for the beginners. So coming from there, this is the web page that Jose is going to briefly walk you through and discuss what the components are and how all these things we are putting together. And I'll let Jose later on to go through it. And next thing, just to give an idea of what kind of information that we have and how we put together this website is, for example, the first step of after data collection is we want to generate the orthomosaics. For that, what we did is we tried to search what are the software that are available to do the orthomosaicing. And we found there are 4 or 5 different software that helps help us to process the raw imagery and generate the orthomosaics. And taking an example, we collected the data sets and then run through these different software and try to do the quality analysis of those software, of the outcome coming from those software. For example, you can see the quality of the orthomosaic in ArcMap here is, you can see some of those spots are not really matching, and going all the way to Metasep, Pix4D, and WebODM. So now another information is Metasep and Pix4D, they are a little bit expensive, and we wanted to see the open source, if there is any open source platform that generates the same quality data sets as Metasep or Pix4D. And we realized that WebODM, which is the open source platform, generates the data of as equal quality or similar quality as Metasep and Pix4D. So these are what we try to put in the web page is we would like to test them and then verify, making sure those information match, and then put those in the web pages. Next slide, Seth. Okay, and additional information when talking about this website is, what we thought is there are so many different programs that are developing their own data processing pipelines, or not coming from the breeding perspective, but from engineering programs or something like that. And they constantly need the data set to test their algorithms or to test their any kind of tools that they develop. So we decided to utilize this website to share all the data sets that we have available for different crops so that they can test some of their tools. For example, someone develops the plot boundary tool, automated plot boundary tool. And if they want to test it for multiple crops, then we wanted to make them available the UAV data sets collected from different crops so that they will have easy test material for their algorithms or for their processing pipelines. And in addition to that, what we thought is, again, going back to helping the community who are just starting UAV in their programs. So we developed a UAS manual. I don't know if you might have been familiar with it, but if not, what we did is we went through the basic steps of what is really needed to collect the data sets, what software to follow, what are the protocols that the software needs to follow to generate the orthomosaics, to generate phenotypic information. For example, canopy height, canopy cover, or vegetation indices. And then all the way getting those data sets in the Excel file for further analysis and comparison. So this is the manual that is available as well, that I think Jose is going to share where you can find or purchase the manual. And with that, I'll let Jose to, oh, okay, Seth, go back.
Either Mahindra or I could talk about this, but I think one thing we wanted to bring to everybody's attention is the third annual Artificial Intelligence in Agriculture Conference, which phenotyping is going to play a role in, but it really covers all of ag, from sociology to economics to agronomy and plant breeding and genetics. But University of Florida hosted last year's, Auburn hosted the years before, and Anna Porter is helping us coordinate this, and I don't think any of this would have been possible without our AG2PI project, and I think they're sort of directly related. So if you have any interest, in presenting either a poster or, if you're from another institution, we could still accept another couple oral abstracts, but we're expecting in the neighborhood of 300 to 400 people for this based on past performance. So with that, I will stop sharing and give the floor to Jose.
Okay. Thank you very much, Dr. Murray. Welcome to the website. This site has been developed for learning, sharing, and exploring the use of UAS in agriculture. Since AI, it's very popular lately, we decided to implement a chat box where you can ask, some questions such as, what is the best time to plant corn in Texas? And you can get it says from March through early A, basically an instant response. In addition to our landing page, we added some learning materials. We added, the basic definition of what is UAS phenotyping. As you might know, in order to fly drones in the US, you are required to have a Part 107. So in here, we added a study guide for the Part 107. And in addition to that, they have many documents on it, but one that I wanted to emphasize is this one over here. This is basically, it's going to help to study, but this guide is also given to you at the time of taking the exam. This, to make reference to maps and everything. So, going through this guide is definitely going to help you pass the Part 107. Going back into the website, as Dr. Murray and Dr. Bandari mentioned, different UAS programs have different phenotyping protocols, and one of them it comes from the Potato Genetics Lab, and they have a very nice compilation of the steps on how to fly, how to connect to the drone, and they even show two different cases for small field or for medium fields, and what they suggest should be the configuration for flying. As Mahendra mentioned over here, we have a link of where we can you could get our manual. This is gonna help you, if you want to learn more about UAS, how to generate your own features, from generating the features up to exporting Excel files with the values. In addition to that, we added a blue UAS clear list. Basically, this list was developed by the government. These are the drones that meet their security requirements. As Dr. Murray mentioned, we recently had a discussion, on what to do when some research labs start to have limitations on what software they can use for SFM. For example, AG2PI Metashape, since it's Russian, some of the labs are not able to use it when they are working with the USDA. In addition to that, what other drones you could be using if you're not allowed to use DJI anymore. So we added links to a compilation of all the slides that were presented during the discussion, as well as a link to the YouTube video. In workflow and resources, we have a list of different platforms. It's the most common ones. One of them is the Free Flight company. Here, the AltaX offers a high payload, and the Astro, it's basically a replacement for the DJI B4R. The same thing we have for different sensors. You can just see a bunch of different options, and for multispectrum sensors as well. For SFM, as Mahendra mentioned, we made a comparison of the different software. Here we included the cost, the platforms where this can be supported, and the output that they offered. Below the comparison table, we have the steps. Let's say that you download a free version of the software. We provide the data sets. You could give it a try with these steps that we have over here. We added steps on how to do SFM in AG2PI Metashape, on ArcGIS, Drone Mapper, and also for WebODM. So once you have your orthomosaics and DSM, the next step is basically to create plot boundaries or plot grid. So one of the tools that we use is QGIS. The reason for using this software is basically you don't need to buy a license for it. So we added the steps on how to create the plot boundaries and the plot grid using this software. In addition to that, there are also more software available in the market, and you can also have a link to them. In this case, Field Image Art not only allows you to create plot boundaries, but also to extract some features from it. We added a list of the most common vegetative indices. And the way it works, basically, once you have your orthomosaics loaded in software such as QGIS, depending on the bands that you have available, you know which vegetative indices you can generate. So here, the way it works, you generate the features, and then with the plot boundary or the plot grid that you have created previously, you just read the values on those vegetative indices. And the next step is basically exported as an Excel file, and then you have the table with the values directly. As Mahendra mentioned, Dr. Jinha's group is developing an automated QGIS plugin to develop plot boundaries. So, for example, for his lab, if they want to start testing the plugin, we made data available. So you can just click on it and it's going to start downloading. I have already downloaded. So let me share to you how it looks like. So for example, for the alfalfa, we have the raw images. In this case, they are all RGB images. In addition to that, we added an orthomosaic, an Excel file with the GCP coordinates, the digital terrain model, which is basically when you fly before plant emergence, and then the digital surface model. So with the DSM and DTM, you can extract plant height.
Jose, sorry to interrupt, but if you want to wrap up, because we're almost running out of time.
Sure, absolutely. I appreciate it, Jack. So we added a discussion forum. You can register, and let's see what else we have. Here we basically, this list gets out, the information gets added automatically, and we don't only have information from iLife, but also different news, basically. And with that, I conclude my presentation. Thank you, Jack.
Okay, thank you very much, Jose, Mahendra, and Seth. Very impressive work, and I think a great example of how the seed grants are helping build community and bring some standardization, which is very much needed, of course. So there already been some questions in the chat. Any follow, and Seth has responded to some of those. Any follow up on that? Or other questions? Just speak up if you have questions, or raise your hand. Otherwise, I have a question. So, Seth, you mentioned that different pipelines give different results, and if you change your pipeline, you may have to redo everything. That's kind of worrying, because pipelines will change, right? I mean, of course, well, hopefully pipelines will improve over time, and unless you want to be stuck in the past, you need to change pipelines. So how much are things going to change? I mean, are results going to very dramatically change that you can't compare back anymore?
Well, that's why our discussion on what data, data standards or saving data, sharing data, stuff like that's really important, because, the orthomosaicing functions still are imperfect. We expect that those will improve, right? So saving the raw images is going to be really critical for later processing. But we also need to be realistic that, processing data currently takes a long time. We're moving on to the HPRC, thanks to Aaron and others' help. But currently, I mean, 20 locations took almost a year of one person's time, right? So you sort of just got to grab a pipeline and stick with it, and not mess with it too much. But that's really the same as genomic data, right? If you remember old trace files, the way that the software called trace files would differ from year to year. And so documenting that's going to be really important. But we don't know exactly how much it'll change.
Yeah. So is there much effort needed to validate? So, of course, you're looking at, well, can you use this data to predict, final yield or senescence and so on? But should we also validate, what we're measuring, but with the drone on that day? How does it compare to a manual measurement of that same thing?
Yeah, I highly discourage that approach, because it sticks you in thinking that manual measurements are somehow correct, when we know what we've seen is, for instance, plant height is just such a great example. There's more error and higher repeatability. There's more error in manual measurements and higher repeatability in UAS measurements, right? So a lot of these new measurements you can't validate. And the way that we validate them is through repeatability measures. Most of us grow multiple replicates. If it's repeatable across those replicates, there's something actually valuable there, right? And so that is our definition of repeatability. And of course, cross-validation is our other way of validating. But I really discourage people from collecting manual measures and trying to match the drone to those.
Yeah. Okay. The question from Christian Tobias for Mahendra, and then we'll wrap it up with this. For small grains with flexible stems in windy sites, are orthomosaics a drawback? And are there any other possible approaches?
I think windy conditions are drawback for almost most of the things that, and in our program, we usually have defined there is a certain limitation on collecting the data sets on if it is windy versus if it is not. And we have seen that, problem in the orthomosaics if it is too windy, especially on for example, wheat, like if it is tiny and it tends to move a lot compared to other bigger stem crops like corn. So Yes, but one of the things that we are more focused towards is not to collect the data when it is too windy, when the canopy is moving a lot. And there are two ways of having those errors. One with the sensors itself, the platform itself, as well as when the plants are moving. So, and Jose, Gina, if you would like to add in on that, feel free to join in as well.
Jose, any additional comments on that?
I agree with Dr. Bandari. I did my research on estimated plant height, and this definitely I saw a lower correlation on windy days because of the plants were moving.
Okay, good. We'll have to wrap it up there for this topic. But if you have additional questions, put them in the chat, and then our presenters can answer in the chat or contact them separately. So we're going to move to talk about data standards and knowledge graphs related to plant stress ontology. And that's a seed grant that went to Dr. Pankaj Jaiswal from Oregon State University. So Pankaj, please go ahead and share your screen.
Can you see my screen with the home page?
We see your screen. We don't. I'm not sure if this is the slide.
Yeah, it's coming up.
Okay.
How about now?
Yeah, great.
Wonderful.
Please go ahead.
Right. Many thanks to the AG2PI team for funding this project and giving us this opportunity to talk to you all. And right now I'm talking from one of the project's goal was to do a workshop hands-on. And so I can let you know that there are about 10 people in the room here, and a hands-on workshop is ongoing. And for this particular project, I'm going to put on my Oregon State hat. So because I'm also a program director at National Science Foundation. So whatever work I'm presenting here and whatever I'm talking is all Oregon State thing. So it's my OSU hat here, right? So we're talking about the plant stress ontology, and many thanks to Seth for telling us a little bit more about the phenomes, and things like that. Those are very critical things that we all are looking at it. But in terms of ontologies, when we talk, is mainly about once you some of these terminologies and vocabularies that we will talk about today are part of the metadata that is being collected as part of your genotyping or your germplasm management or your population management, as well as looking at functional genomics aspects. And the range is anywhere from molecules to population or ecology level or landscape level kind of things that we are looking at it, right? So we are mainly talking about stress, but I'll give you an overview of where the project is, why do we need this kind of a project, what are the ontology 101s, just to give everybody and bring everyone on the same page so they understand what is vocabulary, what is an ontology, and what are the standards. Then I will talk a little bit more about the plant stress ontology and how we are designing it, and what are the few questions that we have heard from our participants in the workshop here, as well as we will likely be hearing from some of the participants here on this webinar. And then I'll talk a little bit more about the broader impacts and collaborations that we would love to seek for this project in general, okay? So as you all can see, and you also, that there are a lot of objects here, and the field is moving. Where is my cursor? Just a second. I'll bring it in here. Let me open up my laser pointer. So a lot of information is being collected based on the germplasm collections in various national and local germplasm repositories or with the breeders, and they have those accessions and populations working on it. But you're also looking at some of the pan-gene set and pan-genome sets that are being developed, and then a lot of functional genomics aspects that are being assayed in order to find the candidate genes and their functions and processes that they work on. But while we get up to the gene level, the breeders and the geneticists are working with genetic markers, loci, and QTLs, and all sorts of other things. Ultimately, all the phenome projects have to drill down to the candidate genes or the region of the genome, which they want to use it for breeding purposes as well. So those are the regions or genetic features have characteristics, and characteristics are in terms of phenomics or phenotypes. But you can imagine those phenotypes can be what is the function of that gene is also a kind of a phenotype, because it's a characteristic of an object. Similar thing is when that phenotype is observed, and in terms of growth stage, or where it is observed in this case. And Seth told us a little bit interesting thing about if you are doing the phenotyping all across your growth and developmental stages, then very likely that you're finding different different candidate genes that may be influential in the early stages of the development that are eventually going to lead up to your final phenotype, not necessarily as many of the plant breeders, for example, would do plant height only at the end of the cycle of the project, right? So there are early events are also critical for those kinds of things. So those are things that when those spatial and temporal aspects are important. And then obviously, what are you observing? That's a trait, and how you observed, and that's a method. And the value and observed value is your phenotype that you're looking at it. But many times these things are also done in terms of stresses, for example, drought or diseases as well. And all these characteristics are dependent on the environment and the condition and the genetic practices or agronomy practices people do use in the field. So there are a lot of factors, and if we don't capture many of these things, parameters, then we are missing a lot of features, and there are holes in the studies. I mean, study may give you a good idea, but it's very hard to replicate unless you have all the metadata available to you for doing, or even to compare one study versus another study in general. So what we are trying to do here is develop some standards on how to record and how to describe some of the features in terms of ontologies that we are looking at it. So moving on to the ontology 101 aspect over here, people are already familiar with a lot of vocabularies people use. Either is a drop-down list in your databases or just a plain list in an Excel or a tab delimited file. You can have different plant structures, plant diseases, for example, or the plant traits in general that's over here. These are the list of the things. But it's very hard to find any connections between these features that you're looking at it. So instead of drop-down lists, people have started using a little bit more structure kind of things over here, as you can imagine, with some relationships, but these relationships are not defined. This is just a plain hierarchy. People will say, okay, a leaf epidermis is just a child of leaf, or leaf is a child of shoot, but there is no relationship. So in terms of generating and querying the knowledge graph or your database, people would not be able to show anything. So if somebody comes and says, show me all the parts of a leaf for which you have identified any phenotype features or your candidate genes that can help me drive my drought tolerance, for example, it will be very hard to use the existing data, which is unstructured in many ways, or the vocabularies or descriptors that are unstructured and have no defined relationships between them. Similarly, we have it for the diseases as well. So that's a step further, but still not perfect. What we are doing with ontologies now, what we take is we take all the terminologies that you saw in the list or in the simple hierarchies, and we started building relationships on those kinds of things. So you can imagine the relationships, for example, in case of an anatomy, is basically a child. It could be a part of something. So think about it that for a disease or for or drought tolerance, there might be several traits that you might be assaying, for example, deep root trait or root length trait, but eventually cumulative effect is they are all part of drought tolerance that you're looking at it. So you can build those relationships in your ontologies, and I'll give you some examples here in terms of ontology. Over here, what you're looking at it, that's, for example, embryo sac is some kind of an organ or a structure, plant structure, and it is part of the ovule that you're looking at it, and then obviously looking at going forward in those things. So relationships are very much defined, and you can actually use these relationships to actually query the data in a much more meaningful way that we are looking at it on that side, right? So that's the ontology structure, and in an ontology, every term that we use, each box is one term that you're looking at it, and every term has a proper name. And it has a stable ID, which this stable ID is never deleted from the database or from an ontology. This is very unique. It's like a social security number that we can think about it. And if the term name can change, but not at all a meaning of that thing, the term name and the stable ID stays as it is. But if the definition changes, that means you're changing the meaning of the term, then the term is retired, but it's not deleted. It still becomes obsolete. So people can still be able to query it, but it's preferred not to use that term at all. There will be a replacement for you. So every term in an ontology has a name, stable ID, a proper definition, which can be read by the computers for doing logical meaning and building other language models, as well as human readable. Every term must have an is a relationship. That's a required thing, and it can have multiple relations. So in a simple hierarchy, you generally have only one parent to child relationships, whereas in an ontology, you can have multiple relationships. Only condition is the child should not become a parent of its own parent. That's you cannot have a circular argument on that side. Many people call the same term by different names, so synonyms are allowed, and comments if you want to highlight some things in general in that way. So with that, how do you use these ontologies in a so it's kind of a graph which we use, directed acyclic graph that we're looking at it. So here's one way of looking at it. If this is an ontology, and you can imagine the blue lines connecting the dots, and they're always moving up, the relationships is moving up, it's is a, and the green ones are part of, and red one is developed. So it says root hair develops from trichoblast. Trichoblast is a part of root epidermis, and root epidermis is part of root, but root epidermis is epidermis tissue type that you're looking at it. So this way you create a knowledge out of this graph that we are looking at it. So for example, advantage of this thing is if a gene cow1 is annotated to root hair or has a phenotype, which you knock it down, there's no root hair development, for example, then it is associated with it. It could be a QTL, or it could be a genetic marker associated with an ontology term. What happens is, based on the relationship, these annotations are accumulated by the parent term, and then you can go all the way up. So it's up to the user if they can enter a resource or this graph or query the graph at a very top level, they will get everything that is accumulated upwards. Or if they know very precisely, then they can come straight away at this level and do the very precise annotations that they are looking at it. So this is a knowledge graph to give you an idea about how the ontology works and how the annotations are worked on that. So this is a standard way of looking at it, okay? So feel free to put in your questions in the chat, or we will have a Q&A afterwards as well. So as of now, the Plantium project that is managing the plant stress ontology is developing and maintaining some of the ontologies that you're looking at it in yellow. That are plant anatomical entities or plant structures, plant cell types, the plant trait ontology describing the traits, not the phenotypes, but the traits. So for example, leaf color is a trait, but if you say leaf color pale yellow, that's a phenotype. So pale yellow is a phenotype characteristic that you have actually observed. What is observable is your leaf color. That's what you're looking at. And then you have crop ontologies, which are very crop specific, like rice crop ontology or maize crop ontologies. These are the vocabularies that are used by communities. And then we try to bring them back into the trait ontology and map them as well. And then you have the plant structure development stages where we describe when and where the certain character or phenotype or a function is observed. On top of that is environmental conditions. It could be a bigger environment, or it could be just a treatment that you have given to your plants. Sometimes those treatments may include some kind of a spray or a pathogen or something like that. And then we have overall covering all of these dimensions is a plant stress ontology, and I'll tell you a little bit in the next few slides about it. So the ontologies in the blue are managed by a bigger consortium of model organisms across the full kingdom, living organisms, and those are very species agnostic, whereas the yellow ones are plant ones, and then we might have a crop ontology is very species level or a crop level that you're looking at, okay? What we do is, in terms of moving into the plant stress ontology aspect, you've seen that there's it could be a rice disease hierarchy or a list where you're describing the fungal or the bacterial or the viral diseases of rice, for example. But when we start building the ontologies, which are species agnostic or even staying species specific, but have some broader level bins across all plants or all crop plants that we are looking at it, then we need to start thinking about some of the aspects of how do we organize the ontology. It could be described by stress type, biotic and abiotic. That is where the plant stress ontology is coming out to be. What is the causal agent that is creating that stress? It could be of biotic origin, which is connecting to a taxonomy term, or it could be an abiotic term for a physical and a chemical form type. So it could be an ENVO ontology, or it could be a biochemical ontology or a chemical name as well. Then we lead to, in case if it is a biotic stress, then obviously it is leading to some kind of a disease or a damage to a plant. So we need to have a nomenclature, access to nomenclature. Was the community is talking about those? How do they use those terminologies and keywords? And obviously, when we're looking at it, there has to be a plant host. And then the host aspect is at what time the agent is infecting, for example, or was sprayed, and at what time the infection is observed. So there are two different levels of this thing. So it's what plant part was infected or sprayed, at what growth stage, and you may see it after a week or after a month, those symptoms appear at a different level kind of thing. So those are the kind of granularity that we need to understand in order to build the knowledge graph that we are looking at. Obviously, the biotic agent, for example, the fungus and bacteria, they have their own lifestyle stages. So you need to take care of what is the spatial and temporal aspects of those work of the biotic agent. And then once these interaction starts happening, then you will see some symptoms anywhere from molecular functions and processes getting affected to the features or morphological features that you can visualize either through naked eye or through your drone images or other hyperspectral images that you're looking at it. So those are the symptoms. And then collection of those symptoms would be your phenotype or manifestation that you're looking at it. So the graph for the stress ontology gets really, really complicated at that level that you're looking at. So for example, rice bacterial disease has a host. Somewhere here would be rice would be the host over here, which is infected. And now you have a plant disease resistance traits that you're observing. And so all sorts of relationships are defined. This was done a little bit earlier, but some of these relationships are now being redefined or reprogrammed in a way to resolve some of the current aspects of the information that we generally have right now. So we are building on those things. So this is an example of a rice, but in the plant stress ontology, we are trying to build top level generic terms and then go along into the species level or crop level that we're looking at. The focus for this AG2PI project is rice, maize, wheat, soybean, some of the tuber crops, potato and cassava, and grape wine that we are looking at it. And then the whole aspect is to do hands-on work, get the feedback from the community, bring the community and experts into the room as we are having it right now here in Corvallis, and try to resolve and get some standards built by the community that can be moved on and start building those structures. So as of now, I think we have close to around 250 plant disease ontology terms into the system, and then we're trying to standardize and go farther to start building. Once we have the pathogen and the disease, we are going to be collecting more on the information on what kind of symptoms and traits that it affects, and what plant parts and other things, so that to complete the whole structure in that way. Okay, so I'll give you some. This is a early development version of the ontologies that we're looking at it. Plantiome project where we are serving this thing. It has lots of other ontologies, but let's focus only on the plant stress one. You can see that there is an abiotic plant stress aspects and biotic. Biotic would have animal damage for grazing and all sorts of other things and herbivory. And then you have plant diseases, also defined across the three other organismal kingdoms over here. And then if you take a look at plant bacterial, for example, then again, you will see that 5 or 6 different crop species specific bacterial diseases are there, and then the diseases. And if you go into the rice one, then you will start seeing some of those. So each of these terminologies would actually fill, for example, rice bacterial disease would have its own unique ID, which never gets retired. As I said, it's a unique thing. And the reason why we use it, let's say a rice base or a rice genotype phenotype database or any of the rice genome database, they can all use the same term and the same ID for the annotation of different objects or defining their experiments. So when you do query multiple resources, you just need the same ID, that features that everybody uses, and the definition stays the same, and everybody allows for that. If there are any issues with the term, then there is a GitHub tracker where you can submit your request either for a new term or edits on the term or definition or relationships that you're looking at it. All the ontologies that we're talking about are maintained in a GitHub, in a central repository that is managed by the Plantiome database. And it's so that it's a global dissemination of all the standards and ontology development and training happens through that effort. And this is a portal for the plant biology, any ontology associated with plants. Once we have it more developed, you will have a more refined definition. So you see the xanthum, its host is Oryza sativa. Its disease is caused by Xanthomonas oryzae. So from the templates or that we are using to build this ontology, and what do you call it as expressions, or what is it called? Design patterns to build this ontologies has the host taxon, the pathogen taxon, and the plant structure aspects coming from the plant ontology to build automatically build these ontologies and the definitions that we are looking at it in general, right? So as we go on, as we develop more, you will start seeing the graph views and the neighborhoods and any of the annotations that are associated with these things. As of now, if you're looking at it, I can expect that it has annotations to germplasm that have been annotated to this disease or disease response. There could be populations, there could be genes, there could be QTLs associated with that, and some of the genetic markers that can be used in general, right? And probably connecting with some of the clinics, plant clinics and pathology departments, then you will start seeing some images of those as well. So some of the questions that are being discussed over here in this workshop that have from yesterday is where do we find all the most updated diseases and pests and pathogen nomenclature? That is one aspect that's I think we're looking forward to working with the APS, American Phytopathological Society, disease catalogs, and as well as the national network of plant clinics. We're trying to find all known symptoms and traits in general. But there are other challenges. There are disease names, same disease names, but different plant species hosts that are coming in. So terminology has to be differentiated. How do you use powdery mildew from corn versus rice versus sorghum? So the nomenclatures have to be assigned and standardized. Same pathogen, but different diseases in different species. That's another challenge that we are looking at it. So the graph would allow multiple relationships. Same pathogen should be connecting to different crops and to the different diseases as well. So that's where the ontology would help. Then again, the same pathogen species, but different names based on the morphemes or asexual and sexual spore forms. That is another aspect that you're looking at it. And so there is community consensus has to be built in. And then obviously I told you about the temporal and spatial aspects of when the infection happened or when the treatment happened and when you actually start seeing and displaying the diseases and symptoms. It takes time from that side. And then another question that we are looking at, some of the diseases are not just one-on-one with pathogen or the environmental or treatment for the abiotic aspects. It could be one-to-many kind of thing. So it could be a complex mixture of pathogens present in that particular disease manifestation that we are looking at it. So it's not very clear on that side. If you go beyond the microbial side, then you end up into nematodes, for example. Some of those reactions and responses are just by calling some kind of a damage, but is it actually a disease or is it a damage? So those are challenges that we are looking at it. How about animal damage is another one. And then there is mimic responses. The abiotic conditions would lead or give you features or characteristics as if they're mimicking a disease response. So how do you define all those things? So these are some of the challenges, but also good questions. And we will be working hard to bring and code those things in the ontology graphs that we are looking at. But the biggest challenge comes out to be is what's after AG2PI funding, right? How do we sustain long-term this plant stress ontology and the other ontologies that are going to be used for building this network in general? So is it sustainable? Where do we seek funding from? And it has to be long-term and a community effort in order to maintain and disseminate this data standards. So that's also a question for this forum as well as for the other intellectuals going around. So here's some of the collaborations and broader impacts that we are expecting. This will be a disease or stress related information that would be presented here, like a disease, what is the biotic agent, how do you mitigate that problem, and what are the phenotypes associated with that. But if you can use this information also to annotate genes, germplasms, and genetic variants and other things, some of this would involve biocuration. Many of it can be automated to build this reference ontologies, and the data would come from many other databases in general. But for disease and stresses, I think it's the APS disease catalog or National Plant Diagnostic Network of plant clinics would be instrumental in providing us the data, as well as we working with them directly to cross-link the data sets that we have in general. But in much larger and long-term prospects that you're looking at it, once you have this knowledge graph, you can build different data layers and tools to actually help ag extension and stakeholders as well, because these would be the first responders who would be actually detecting some of these things early on in their fields. How can they detect and then start looking for the information so that you don't have to put extension agents on every ground, right, every other place? Can you make it as a digital aspects and then mitigate those problems as well? So all of this data would be plant stress ontology is being presented out of the Plantiome portal that you would look at. This is the centralized infrastructure that we have created for all ontologies related with plants and plant biology. And it's a global effort along with the Biodiversity and CGIAR Alliance that we have here. And we work diligently with all other ontologies that are developed by the biology community or OBO Foundry or biomedical community that we are looking at it in general. So with that, I would like to acknowledge some of the funding that's provided by the AG2PI through NIFA. Small funds come from USDA ARS to maintain the plantium site, and my time and early development of the plantiomes was all funded by the NSF plantium team that we have, and the CGIAR Alliance Biodiversity at CIAT, Elizabeth and Murray Angelique. And a lot of bioinformatics help comes from Chris Mungle. And some community efforts and outreach through Graham King and DipSeek that we are looking at it. And obviously we have about 10 people here in this current workshop that we have. So with that, I would like to end and thank you and take the questions if there are any. Thanks.
Great, thank you very much. Very very informative, very impressive, and you know, I can definitely see the importance of this work. There are some questions in the chat, and please keep adding them. So Edwin Reddle asks, are these ontologies BR API compatible?
Yes.
We focused on trait method and scales.
Right. Yes. Once the ontologies move away from the development version and go integrated into the Plantium ontology database, then you will be able to access all these ontologies through the standard APIs that will be coming in through. Plus, many of these ontologies that we are developing are also distributed through ontology lookup services and OBO Foundry tools. They have their own APIs, as well as I think AgroPortal is another one in France that serves. So it's not just served out of our database. The raw data files or ontology files are picked up by many other resources around the world, and they all also serve it through their own APIs as well. So yeah. Absolutely.
Okay, great. Another question is the crop ontology community, which includes about 40 crops, expected to cross-reference all biologic stress-related terms to the stress ontology. So for example,
so early on, I think the biggest bottleneck when we were writing this AG2PI proposal was that anatomy and morphology traits and quality and yield traits work out pretty well with the reference trait ontology, but there was not a whole lot of enrichment of plant stresses and plant diseases. So with the development of the PSO, we expect that almost every term, that's the goal, is every term in the crop ontology must have its equivalent in the reference plant stress ontology, so that we will be able to map it in general, right, as well. Yeah.
Okay. Seth, go ahead.
Yeah, Pankaj, I mean, I love that you work on this because it's so important and so many of us don't think about it. I do wonder, as we start thinking about new phenomic traits that may not be as well defined or as well characterized, I mean, will this require a rethink, or do you think that they'll just plug into the existing framework?
No, I think it's a very good question, and we have been discussing about it, how to go about it, because many of those are digital traits that we are looking at it, like what the gene indices, or rather, or vegetation indices that you already have, right? So NDVI and GNDVI are basically defined, but there are other 21 or 61 other vegetation indices that you identified. So we will have to come up and work with the community to see how do you name them, how do you define them, and what is the methodology behind it, so that everybody is on the same page looking for it. Absolutely, these are all digital traits, which are not the regular plant traits that are being used by the plant breeders earlier. So a lot of thinking has to go in. Absolutely.
Yeah, I was thinking the same thing. That, you know, listening to the earlier talk, you know, there's a deluge of additional phenotypes and traits that are going to be used, and
absolutely,
it's important to have, I think, to have the framework in place like you have, and then see how you can incorporate those. Yeah, so.
Absolutely. Yeah. And some kind of an enrichment, and some traits like PCA based or things like that, or machine learning based traits, or large language model would find some new features that people have not ever thought about it. So those are challenging, but absolutely, it's a long-term goal.
Yeah.
To bring them in.
Yeah. Other questions? Just raise your hand or just speak up. I have another. So I'm a little bit familiar with the trait ontology project on the animal side.
Mhm.
I'm not familiar enough with it to be able to say that what is done there is similar to what you're talking about. But are there a lot of similarities on the different ontology projects for the different biological systems?
Yes, what many of the model organism databases have phenotype ontologies for them. So think about it that, as I told you, that when we were starting to collect and curate the plant data from different crops, it was very difficult to compare and annotate everybody's methods and scales and variables for phenotyping, right? How do you convert the numerical scales into your kind of qualitative scales? Or even to compare somebody's counting from 0 to 9, the same other guy was counting from the reverse order. As well. So comparison at that level was difficult, especially in the plant community. Model organism community is pretty small, so they all use the same vocabulary, so it's pretty easy and going around it. So that's why we in the plant community we stayed a mile above. At least at the trait level, our language and our concepts that we are observing are more or less the same. Leaf color is leaf color. How you measure whether it was a yellow or green or red or pale yellow, those are very detailed level scoring mechanisms that people do. So at that level, there is no harmony across within the a single crop or across the crops in general. So the trait ontology for plant trait ontology has stayed more or less at what you are trying to observe at that level. And that has been the practice. Animal system has gone up to including the phenotype values and scores as well. That's the only major difference. But at the top level, I think we all have morphology, anatomy, quality, and growth stages. And then obviously the stresses. At the top level, they are more consistent in the binning levels. Yes.
Okay, good. Other questions? Either for Pankaj or for the group from Texas A&M? Okay, if not, Pankaj, you want to stop sharing your screen, please?
Yes.
So yeah, I want to thank all presenters. I think it's great to see how the funds we were able to distribute from NIFA are being put to very good use, and they'll definitely have an impact years to come and lead to additional work. Couple of announcements before we wrap up. Yeah, we will have another field day in March 20, and then another one in April. There will be some additional workshops, and we'll send out announcements on that fairly soon. Couple of other things I want to draw to people's attention. One is that there is a new RFA from AG2PI. So starting next year, that is managed directly by NIFA. So there's another 2.3 million that is available. Deadline is May 30. Program leaders Christian Tobias, who's online, and Angelica van Goor. And then there will be a technical webinar on that RFA, March 21. And I think Nicole has sent out the URL to register for that. So definitely, if you're interested, please register for that webinar. And yeah, keep the submissions going. The other thing I should mention about that, in relation to that, is that, of course, Congress and Senate, they are still working on this year's budget, and it's important that AG2PI funding enters that budget again, ideally at a higher level than we've had so far. And also the new farm bill is being discussed, and again, it's important that AG2PI is continued in the new Farm Bill at the 40 million that I think that's the request, that it enters again at the 40 million that was per year that was entered in the previous Farm Bill. Of course, we didn't. That wasn't appropriated, but it's important that it goes into the Farm Bill again. So talk to your experiment station directors and talk to your legislators leaders about the importance of AG2PI and how you've benefited from the funding that has been made available so far, and so we can make sure that it continues and expands. Last thing I'll mention is that there is one of the seed grants, and was also a coconut grant, and an awardee for in the last round through NIFA is a project that I'm also involved in on data sharing. PI is Hao Cheng from UC Davis. So this is about data sharing and protecting confidential and proprietary information. And we will have a workshop on that at the upcoming AG2PI Ag meeting, which will be in Phoenix, April 15 to 17. I now realize that it overlaps with the AI and Ag meeting that Seth talked about. Yeah, which is unfortunate, but that's the way it is. But anyway, so April 15 to 17 in Phoenix, they will have a workshop. For that meeting in general, if you're interested and you haven't registered yet, the early bird registration ends March 5th. And if you're able to get your abstract in today, the deadline for late breaking abstract is the end of today. So look at that, AG2PI.ag.org, I think is the website. So look at that. With that, thanks again for all the presenters and for the participation, for everybody who was online, and hopefully this was again useful. And if you have any comments, I think there's going to be a survey, Nicole. So as you exit, we will ask you to fill out a brief survey. I need to thank Nicole Scott again for all the logistics, and also Eddie on the mechanics of the website and so on. That is continuing to be maintained, although our funding has ended. One more thing, we do have some additional funding left for student travel awards. So if you have a student or if you are a student who is interested in presenting something at a conference that is relevant to AG2PI, please apply, and it's on the AG2PI.org website. So with that, thanks all, and hopefully we'll see you next month.
Thank you, Dr. Doctors. Thank you, Dr. Scott.
Thank you, everybody. Bye-bye.
Thank you. Bye-bye.
Bye.